Hello, I'm Ryan, and today I'll be giving you an overview on CAR T cells. This presentation will be split into three main parts. First of all, what is CAR T? Then, how is CAR T made? And lastly, why do we need CAR T? Firstly, let's talk about the bloodstream. Our blood has three main cells: the red blood cells, white blood cells, and platelets. The red blood cells carry oxygen and carbon dioxide around the body, and is what makes our blood red, while platelets form clots to prevent bleeding and is what makes your scabs. And our main character today, the white blood cells, are part of the immune system, which kills antigens and pathogens, which are the things that will harm your body. Now let's go more in depth into these white blood cells. As said before, white blood cells are the guardians of your body. It is a cell that specializes in destroying antigens, the dangerous things in your body. This includes bacteria, viruses, and cancer cells. Without the white blood cells, the organisms can get infected easily, and that includes you. And there is nothing to stop the danger from hurting our body. And we can die very easily without them. Now you may wonder, what exactly are these cells? Where do they come from? White blood cells and red blood cells and platelets, they all come from what is called the blood stem cells, a cell that's produced in your bone marrow. The blood stem cell will then specialize into myeloid stem cells and lymphoid stem cells, and these can then develop into red blood cells, platelets, and various white blood cells. As seen in the diagram, everything encircled by the blue square, those are all different types of white blood cells. Now that you have a basic understanding of the cells in our bloodstream, let's talk about our main protagonists, the T cells and B cells. T cells are a kind of white blood cells, and they specialize in wiping out the infected or cancerous cells, while B cells are another kind of white blood cell, and they specialize in creating antibodies to remove antigens from your body. These two are important because the T cells are what will become CAR T cells, and B cells are usually the biggest problem that we have to face. This is because the main patients who need CAR T are those diagnosed with B-cell leukemia or lymphoma. This is basically a B-cell cancer where abnormal B cells have mutated. These mutated B cells won't be able to be identified by the T cells and will use up lots of resources and space that other cells may be able to use instead. This will cause the body to be harmed, as a lot of the resources are being used on these useless mutated B cells. The CAR T cells will be able to find what these T cells cannot, which are these mutated B cell cancer cells. With these CAR T cells, your body will be able to correctly identify the cancerous cells and eliminate them before they can cause any more harm to your body. So I've said CAR T many times now, but what exactly does CAR T stand for? CAR T stands for chimeric antigen receptor T cell, and since it's a form of immunotherapy, we also add therapy at the end. So chimeric stands for artificial or man-made. Antigen means the bad stuff in your body, so in this case cancer. The receptor is the proteins that can bind to other cells, and in this case is the receptors on the CAR T cells that we are adding. T cell, as said before, are the white blood cells specialized in killing cancerous cells, and therapy just means treatment. So all in all, CAR T therapy just means a man-made T cell with receptors that can bind to other cells that can kill antigens, which are the cancer cells in your body. And this is a treatment that will be able to heal you. So now that we know what CAR T stands for, what exactly does it do? So basically, in your body, you have a ton of cancer cells that are growing and multiplying. These are very bad for your body as it takes up resources and space. Now, when the T cells come over to check on the cells, they will see just a ton of cancer cells just multiplying and multiplying and multiplying. And when the T cells look at this, they think, yeah, that seems about right, because the cancer cells are concealing themselves from the T cells. However, we don't want this because we need to get rid of the cancer cells. So we take these T cells and we turn them into CAR T cells. And when we put the CAR T cells back into your body, the CAR T cells will look at these cancer cells and go, oh my god, this is completely out of control, and proceed to eliminate them. This is the importance of CAR T cells, as they are the ones that will actually be killing your cancer cells. Now that we know what CAR T cells are, let's talk about how to make CAR T cells. CAR T cell manufacturing can be split into 8 main steps. First, receiving the apheresis product. Then enrichment and selection, activation, transduction, cultivation, harvest, getting the final product, and sending the CAR T cells back to the hospital. We will be going in depth in each of these steps in the following slides. But first, we have to talk about our most important pieces of equipment, the Clinimax Prodigy. The Clinimax Prodigy is a closed system that moves lots of tubes and valves around to make our T cells into CAR T cells. We'll put all our materials onto or into the machine, and after a few days, we can extract our CAR T cells from the cultivation chamber of the Prodigy machine. On this slide, we can also see some other machines, such as the tube welder, a balance, and a biosafety cabinet. Now that we have a basic understanding of what is needed to make CAR T, let's start talking about how to make it. Making CAR T cells here at HKIB requires 12 days of cultivating CAR T. But first, we start with day 0 before we start manufacturing CAR T cells. Day 0 is the day where we prepare all the materials that are needed for creating CAR T. The steps included are receiving our apheresis product, enriching and selecting our T cells from the apheresis product, and activating our T cells. First of all, we receive our apheresis product. This is our blood sample. In this blood sample, we have tons of white blood cells that are given to us from the hospital. These white blood cells are extracted from the patient's body, so include the patient's DNA too. This apheresis product includes the T cells that we need that we will turn into CAR T cells. Without these T cells, we won't be able to modify them into CAR T cells, since CAR T cells are something that we modify into, not something that we just create out of random chemicals. Now that we have the apheresis product, the next step is to enrich and select our T cells. This is basically the step where we filter out all the cells that we don't want and we only get our T cells. As said before, in our bloodstream we have tons of red blood cells, platelets, and other white blood cells as well. But we only want T cells, since T cells are what will become CAR T cells. We will take the apheresis product and we put them through a filter so that we filter out all the cells that we don't want, which are the red blood cells, platelets, and other white blood cells. And all we are left with are the T cells that we want at the end. Then we have to activate and cultivate our T cells. T cells have to be activated in order to multiply, as in the body, activated T cells are the ones that can proliferate and attack antigens. However, for us, we don't activate the T cells for it to attack anything at the moment. We activate them because we want the cells to multiply. And since we will eventually have to make CAR T cells and put them back into the patient's body, we'll need enough of these T cells to turn into CAR T cells. And so activated T cells will be able to multiply and let us get a sufficient amount of these T cells. We also want dividing cells because dividing cells make transduction easier. Now you may wonder, what is transduction? Well, transduction is the step where we actually turn the T cells into CAR T cells. Now after day 0 is day 1. Day 1 is the day where transduction actually starts, where CAR T cells are finally being produced. On day 1, the main steps are transduction and cultivation. Transduction in a nutshell is basically inserting the chimeric antigen receptor genes into a T cell to turn them into CAR T cells. At HKIB, we use lentivirus to modify the genes of the T cells. The first step is to obtain our lentivirus. It is a man-made pseudovirus that contains the CAR genes that we need. Then we mix the lentivirus with the T cells, and so the lentivirus will insert their CAR genes into the T cells. And the T cells, armed with the new DNA, will grow out new receptors. And these receptors will allow them to be able to identify and destroy the cancerous cells. And these will be our CAR T cells. Keep in mind that the CAR T cells are just modified T cells, so they still have the original genetic code of your T cells. Therefore, when placed back into your body, it will be seen as one of the body's cells instead of just some random machine that came in. This is also why CAR T therapy is also known as a living drug. As transduction occurs, we also have to cultivate our T cells and also the newly made CAR T cells. This step is basically turning one cell into more cells into even more cells. This process takes 5 to 12 days, which is also why our CAR T manufacturing process takes 12 days. After day 1, day 2 to 4 are just cultivating the T cells and CAR T cells so that we have more and more of them. On day 5, however, the food should be used up as the T and CAR T cells have been multiplying a lot and using up all the food. And the waste bag should be full as they have been also excreting waste. So on day 5, we'll have to replace our food and waste bags for the manufacturing of CAR T cells. We will also have to take out some samples of the current CAR T cells to see if transduction is occurring at a correct rate. After day 5, we just continue to cultivate our cells until day 12, the last day of cultivation. On day 12, we will harvest our cells and prepare them for the hospital. We extract the cells from the cultivation chamber of the Clinimax Prodigy machine, and these cells are now CAR T cells that we'll be able to implant into the patient's body for treatment. However, the cells will die quickly without the proper environment, so we have to use the final formulation solution to preserve these T cells for another 24 hours before it gets injected back into the patient. These CAR T cells will be put and blended into the final formulation solution, which is made of salt and human serum albumin, which will be able to allow for the cells to live as they get transported to the hospital to be sent back to the patient. And finally, on day 13, the day we extract all our CAR T cells, we transport all our CAR T cells into the hospital, which will then be injected into the patient for treatment and healing the cancer patient. The CAR T cells will be able to go into the patient and correctly identify cancerous cells and kill them off before they can cause any more harm. Now lastly, why is CAR T important? Why can't we just use some other therapy such as radiotherapy or chemotherapy? Well, first of all, CAR T cells allows for the body's own immune system to fight back against the cancer, effectively making it a living drug. And it also lets the T cells recognize the targeted cancer and prevent the specific cancer from repopulating for a while. And this treatment can also last many years. Lastly, there's also a very high chance for the treatment to be effective and doesn't have as dangerous side effects such as from chemotherapy. However, there is a chance of relapse, meaning that this cancer might still be able to come back after all the CAR T cells have died off. And also, this treatment is very, very expensive, as it can cost millions and still has some side effects, so it should still be a last resort. Thank you for listening, and I hope this helped you understand what is CAR T cell therapy.